My name is Xinhu Yu. I'm the chair of RMIT Professorial Academy. This occasion is one of the very important tasks of this academy is about promoting the excellence of research, education, and engagement, and also giving university advice and engage with industry as a forum. So today is actually it's my great pleasure to introduce Professor Elena Ivanova. She's going to present the fourth RMIT Distinguished Lectures. Just before I introduce her, just give you a bit of introduction of the RMIT Professorial Academy. This academy was established by the Vice Chancellor in 2017. The purpose of that is basically trying to make use of the brands and influence of RMIT distinguished professors, which at this point in time we have 18. Use them as a think tank to advise the university on future strategic directions, as thought leader, and also as advocate on behalf of university. So the lecture is one of the important elements of the whole of that mission, and we will do more than just that. In the future, we also will hold a public forum on issues, on strategies, on policies, which I think you are more than welcome to participate. Okay, today's talk is presented by distinguished Professor Elena Ivanova. She joined RMIT 2017, I believe. 2018, right. She's from Swinburne and one of the high flyers. Today's talk is about superbug. I think that's why I think this is one of the highest number of participants, just because of interest. But I think it's more than because of the interest of the topic, and also I guess people want to know you more about what are you going to do for us. So she's going to talk about her research and also her perspective, more than just her own research, a perspective about the future. What do we see the future? Certainly we all worry about superbug, so that's why we're all very interested to hear what you have to say. So please give a round of applause to welcome Professor Ivanova.
Thank you very much for a nice introduction. I'm okay? No echo? I'm very honored to talk to you today about our work. And as you see now, it's one of the major concerns worldwide epidemics of superbugs. And I tell you that we are not losing the battle. There is a light in the end of the tunnel, and we'll see how we can do it. As an introduction, I would like to talk about some problems imposed by antibiotic resistance, bacterial infections, and actually emerging superbugs. And then we consider how insect wings, natural bactericidal surfaces, could be a potential solution to some of these problems. And of course, I'll show you examples of novel mechanical ways how we can kill bacteria without any need of antibiotics. The rise of superbugs. I'm sure you're all well aware of increasing numbers of reports telling us really dramatic stories. Just a few of them on a board. Say 2017, the Center for Disease Control and Prevention reported that a woman in Nevada died from an infection resistant to all available antibiotics in the United States. In the same year was another report in China when superbug, Escherichia coli, in this case, was found resistant to last resort antibiotics, carbapenems. That's the antibiotic of broad spectrum of beta-lactam class. And in fact, in Victoria, 2016, 56-year-old man from rural part of Victoria with no history of hospital contact or international travel was reported to have died from Klebsiella pneumoniae, and that particular strain was resistant to all antibiotic available in Australia. Just to tell you that the background of this slide, what you see here, it's an electron micrograph of Staphylococcus aureus, or golden staph, which is resistant to methicillin, and you see how it colonizing, how it can colonize the blood, some blood cells. It's growing and colonizing the cells. Now, before we going any further, I thought it will be useful to give you some terminology so that we all know what is actually a superbug. A definition I found in the Royal Institute of Victoria 2017, a superbug is usually defined as a microorganism that is resistant to commonly used antibiotics. And of course, antibiotic resistance, if you look at Nature website, you find a definition of antibiotic resistance, which is an ability of microorganisms, and that could be bacteria, fungi, protozoans, to grow despite exposure to antimicrobial substances designed to inhibit their growth. You may ask me how we now come to this point where the antimicrobial resistance is developed. There are several different ways. The major reason is selective pressure through drug use in medicine. Another serious reason is that uncontrollable use of antibiotics is in many countries. Australia is not there, not in that list, and also inappropriate prescriptions. But the major misuse is in agriculture. It's up to 70% of all antibiotics produced worldwide actually used, as you can see here, mostly as a prophylactics against disease or as a growth promoter. And of course, we do not forget about genetic transformation, where the genes with developed resistance to antibiotics could be easily transferred amongst different groups of bacteria. Again, you may ask me, is it so difficult to kill bacterial cell? It's so small. Well, look at these facts. Temperature, high temperature can destroy proteins, nucleic acid, and damage membrane, but yet we have bacterium called Paralobos fumari, which can grow at temperature up to 113 degrees. How about radiation? Radiation can kill bacteria? Yes, it can, but not all of them. There is a bacterium called Deinococcus radiodurans, which can withstand ionizing radiation up to 20 kilogray, right? And also UV exposure. What about pressure? There are actually obligatory piezophilic species that can grow at 70 to 80 megapascal. What about reactive oxygen species? In fact, oxidative damage induces production of antioxidants and detoxifying enzymes by bacteria, so they can easily develop resistance to any reactive oxygen species. Now you can ask me again, why they're so smart? Why the bugs are so smart? And I'm always saying that don't forget that bacteria are the oldest life form on our planet. They survive evolution of 3.8 billion years. And of course, they develop versatile metabolic pathways. They can colonize each and every surface. They can survive everywhere. Now look at this example. It was estimated that the total number of bacteria in 70 kilogram, so-called reference man, right, could be 38 trillions. 38 trillions. It could be roughly up to 5 kilograms we're having in our body. Look at this scanning electron micrograph, which I use as a background of these slides. What you see here is a community of bacteria which are found on the surface of our tongue. Right? So bacteria are everywhere, and looks like it's not that easy to kill them. Now, how do they survive? You may ask me again. Of course, a single bacterial cell will not be able to survive because it's very tiny, it's not protected, it's very difficult for it to survive. What happens? Bacteria survive in communities, and these communities called biofilms. What you see here is just a single bacterial cell settled, they call it free-floating planktonic cells, settled on the surface. They aggregate, form microcolonies, and secrete extracellular polymeric material. Right? That's how the biofilm is formed. The biofilm growing further and producing a complex three-dimensional structure, something like that, what you see here. And cells there protected by extracellular polymeric materials against host immune cells and antibiotics, right? So when the biofilm reach a critical mass, the cells could release, planktonic cells could release again and colonize each and every other available surface. That's how the bacteria spread once they form a biofilm and released from the biofilm. And this is also a reason why they can easily develop resistance to antibiotics, because in the biofilm they are protected, right, from any antibacterial treatment. In fact, and they form, of course, infections. Implant-associated infections is #1 cause of implant failure in patients. What is also dangerous is that once the biofilm reaches the bloodstream, bacteria can colonize any moist surface in our body. On this little picture, you see sites of primary and secondary infections. And in a way, that was our motivation. We wanted to design surfaces which will be free from bacteria. So we wanted to fabricate surfaces which could be used for bacterial implants, right? What you see here is a little movie made in our lab, maybe a good 10 years ago now, showing how quickly golden stuff can colonize the surface of titanium, which is commonly used implantable biomaterials for the hip replacement, you will see here. And here is a real scanning electron micrograph of the biofilm form Staphylococcus pseudointermedius on an orthopedic bone screw. That's what is happening. Now, as I say, our motivation was to design these surfaces, but how we rationalize our motivation. We were thinking that because bacteria survive through colonization of surfaces, why don't we look at the surface topography and see if we can modify the topography of the surface and stop bacteria from attaching. Once we stop bacteria from attaching on the surface, the biofilm will not be formed and we will be safe. So topography was our focus. And the very first experiment we've done, but before we go there, I tell you currently what are traditional approaches where people try to design antibacterial surfaces. Of course, we are not the only one group trying to design antibacterial surfaces. Currently, there are basically two major classes of antibacterial surfaces. Antibiofouling, which will be repelling bacterial cell from the surface, and bactericidal. In this type surfaces, the bacteria in contact with the surface will be killed effectively. For that, we use a low molecular weight antibacterial compound, including antibiotics. It could be antimicrobial peptides or even silver. Unfortunately, there are some drawbacks associated with these approaches. Toxicity, non-uniformity coatings, decrease in efficiency, environmental health concern, and of course, microbial resistance to antibacterial agents. So back to the start of our work, what it was now many years ago, and the attachment point or settling point theory was one of the main well-accepted dogma. What it says is that the organisms smaller than the scale of the surface microtexture will attach in larger numbers in micron scale shelters on a textured surface and will have greater adhesion strength because of the multiple attachment point on the surface. So we thought, all right, how about very smooth surface without no attachment point? Would it repel bacterial cells? And the very first, very simple experiment was done one of our PhD students. is just using the standard microscopic slide, which is actually quite smooth with the average surface roughness in the range of 2 nanometers. And after the treatment with the buffered solution of hydrofluoric acid, these little peaks are gone, and you see the surface even more smooth with this 1.3 nanometer roughness. What you can see here is just a remarkable response of bacterial cells, where you see that the cell numbers on the nanosmooth surface, which is really not much changed, is even increased, right? Also changed morphology, morphological transformation, and extracellular polymeric substances. So we reconfirmed this result using different surfaces, including titanium, and also using different range of surface roughness, and the results were the same. As now, the main conclusion coming from this work is that the bacteria are far more susceptible to nanometer scale roughness than previously believed, and nanosmooth surfaces do not represent a repelling environment for bacterial attachment. So that's why we had to move on and find any other surfaces which may be free from bacteria. And of course, we always look at nature. In nature, what you see in nature, there are lots of examples of bacteria-free surfaces. And these are plants or insects where you can find them free from bacteria. Now, that's the particular self-cleaning effect associated with this type of the surfaces, and it's mostly due to the superhydrophobicity of the surfaces, which in turn is a combination of surface chemistry and a particular nanotopography, right? So these superhydrophobic surfaces, in a way, not wet, right? And the water droplets just bouncing on these surfaces and sliding, rolling off. And on the way of rolling off, it takes all contaminants away from the surface remaining clean. Now, what we've done next, in collaboration with our colleagues in Laser Center Ivanova University, we were able to mimic the surface of lotus leaf on titanium, and that's how it looks like. On your left-hand side, you see an example of water droplet behavior on a glass surface and on a superhydrophobic surface with the water contact angle of 153 degrees. Now, what happens when we immerse this surface in suspension with bacterial cells? And for that, we use two different types of cells, Pseudomonas aeruginosa and Staph aureus. You see, Pseudomonas aeruginosa didn't really attach on this surface. However, Staph aureus quite successfully colonized this type of the surface. Now, we had to answer the question why. And that was a brilliant work of our PhD student at that time. Trying to move on. Yeah, probably you've seen it's Dr. Truong, now also a staff member of School of Science. So what we looked at what happened to surface when they immersed in water. What you see here is a little movie showing you how quickly air replaced by water. Right? So, and that is really followed by the aggregation of Staph aureus cell and their colonization of the surface. Basically, air was one of the most important components in the surface, and the surface lost air, and it became favorable for attachment of Staph aureus cell in particular. So that was quite disappointing result, right? So we looked at other examples of bacteria-free surfaces in nature, and that are insects. So the next object for our work was actually cicadas. You all know that cicadas are the most loudest insects in the world, and there are numerous species, and in the range of 200 species are available in Australia. So we used in our work one particular species called Psaltoda claripenis. If you look at the nanoscale wing of the cicada will be composed of a tiny nanopillars of just only 200 nanometer tall and 60 nanometers in diameter. It's a very fine pattern which formed in the epicuticle of insect. What you see here is really the wettability map constructed by another PhD student showing that the surface maintains high superhydrophobic feature with the water contact angle ranges from 163 to 173 degrees. What happens with bacterial cells when we immerse membrane of insect wing in this suspension with bacterial cells? And as you can see, we didn't get the result which we expected. We thought that bacterial cells will not be able to attach on the surface. They will be repelled, similar to the water droplet. But what you see here, it was quite the opposite. Bacterial cells happily attach on the surface, but looks like they're not so happy, right? So what you see here is a collage of scanning electron micrographs, IFM scans, that's also FIBACM, and confocal imaging, indicating that the cells actually dead on the surface. In other experiments, this atomic force experiment, what we show that the bactericidal efficiency or the killing process of the cell on the surface of the wing is remarkably fast. In the range of 200 seconds, you see that we detect a rupturing point. And it was quite a nice match with the height of the nanopillars of 200 nanometers. So basically, one single cell could be ruptured in 200 seconds by, and I'm talking about Pseudomonas aeruginosa cell, a particular type of the cell. The efficiency, the killing efficiency, also quite good. What you see here is that the number of viable cells remaining in suspension was decreased by 96% per square centimeter over 30 minutes. So that was quite, we were quite happy with the results. Now the question then again, how this happened? Was it a chemistry playing a role in this activity or not? In order to eliminate the effect of, or understand whether or not chemistry has any effect, we spattered gold on the surface of the wing, and it's a very thin layer of just 10 nanometers gold, so that the pattern is not changed. So we wanted to maintain the pattern, the same type of the pattern. And what you see here, that in fact the bactericidal effect remained the same. And the conclusion for this work was that the surface chemistry is not really important in bactericidal effect of cicada wing, but rather it is topography that is a dominant factor. The question, another question is to ask how it's happening. What is the killing process? What is involved in this killing process? The theoretical analysis done in the in collaboration with a group of physicists in Spain, in University Rovira i Virgili. That was a PhD student, Sergei Pogodin, at that time. He modeled the membrane of bacterial cell as elastic layer because the thickness of the membrane in the range of 6, maximum 10 nanometers, right? And the diameter is quite large compared to this one. And what really happens in this particular situation? The bacterial cell is suspended on the array of the nanopillar, and the stress which is developed upon this suspension such a high that it breaks, but it breaks rupturing in between the nanopillar. So in a way, the pillars cannot pierce the cell. And this is very common mistake when some of the people trying to understand what happened on the surface. It's all about dimension. So the large like this pillar, 60 nanometers pillar, cannot really pierce a very thin layer of the membrane. Rather, it stretches and breaks in between the pillars. Unfortunately, this particular type of the nanopattern is only active against gram negative bacterial cells. Gram-positive cells, and we tested quite a few of them, gram-positive bacterial cells remain safe on this type of the nanopattern. The reason for that, we believe, that's our hypothesis, we didn't prove it yet, in the different thickness of peptidoglycan layer, which adds for gram-positive bacterial cells additional rigidity for the cell, so it's much harder to rupture it. Right, so that's our working hypothesis. Hopefully, we'll try to prove it. Now, the next object in our work was dragonflies. Dragonflies, another numerous group of insects. And if you look at the pattern of this insect, it's quite different. That is the pattern of the dragonfly. Maybe it's not that clear here, but it's pretty much clear here. It's very random array of different lengths, quite some different lengths of the nanopil. It's very difficult to describe it. But what is interesting here, this type of the nanopattern is very active. Basically, it ruptured all cell types, including Bacillus subtilis spores. And this is quite remarkable observation. So we wanted also to understand if different types of, or different species of dragonfly have exactly the same pattern, but looks like they are not, and whether or not they are the same. Well, what is exactly the same? It may be visual similarity. If you look at the surfaces of the dragonfly wings, they look quite similar, but yet they are not identical. On this little movie, you see the behavior of a water bouncing taken by a high-speed CD camera, which takes 28,000 frames per second. So that's what you see here. That's how the water droplet feels when it bounces out of the surface of dragonfly wing. And on this table, you see the comparison of the nanopillar height and density per this studied species. Quite similar, quite similar. But when we use 3 different species of dragonfly and look at the bactericidal activity, it appears to be different, right? So the that's what you see on this slide, that the one, Diplocoides bipunctata, is the most active one, and the 2 others which we use is quite different. IFM topology analysis showed that, in fact, the curvature, the geometry of the pillars may be the key when the slight variation in the nanopattern may severely affect the bactericidal performance of the nanopatterns. Our next work on trying to understand using a the forces which are involved in the killing bacterial cells. And this work was about the simplified model of cell membrane, which is really the giant unilamellar vesicles or grooves, right, which were constructed from DOPC, and we used 2 other species, different species of dragonfly. One, just a little bit. Stacking. One species, probably the battery is not moving. Or it's the software stuff. So on this image, you see a cryo-ECM image of the of the goof, which actually was done for the first time. For some reason, despite that, that's a very common model of liposomes construction from very well-defined lipids. We couldn't see that before in a cryo-ECM analysis. So basically, what we've done at that time was the understanding whether or not these giant goofs will be ruptured on the surfaces of both species of the dragonflies. And in fact, they were both ruptured on this type of the surfaces. And that's also, that's fine, beautiful. So what you see here is, thank you very much. What you see here is a confocal scanning electron micrographs and cryo scanning electron micrographs showing different stages of rupturing, of grooves rupturing on the surfaces of the wing. And that allowed us to estimate that the minimum amount of additional tension required to mechanically rupture the grooves was in the range of 4.3, 6.75 millinewton per meter, right? But it's all dependent on the relative size of the grooves. That was pretty good match with the previously reported data obtained using a micropipette aspiration. So we were pretty happy with that, but that's not a direct estimation of the forces. It's yet something to study further. So that's a lot of work to be done. Now, natural bactericidal surfaces are very good, very interesting, but the question is, can we fabricate their synthetic analogs? And the answer is yes, we can. What you see here is an example of first biomimetic analog, as we call it, of a natural bactericidal surface called black silicon. In fact, it's quite easy. The fabrication of this type of the surfaces is quite easy. It's a matter of optimization of the parameters. So I'm not going into technical details of the fabrication, just only tell you that it's basically a plasma-assisted reactive ion etching, quite common technique. Once again, it's a matter of recipe and optimization of the recipe, and you can quite reliably and consistently get the same pattern of your choice. Now, on this slide, what you see is a similarity of the pattern which we found on the dragonfly wing and black silicon. Once again, what you see here, it's quite similar, but it's not identical pattern. And in a way, it's a good thing, which means that the pattern which will be bactericidal may not be necessarily identical to the previously found or designed or fabricated. It could be different, but of course the variation may be in a very narrow range of the nano features. So that's what you see here. That's a dragonfly, and this is a black silicon pattern. The table gives you an overview of the bactericidal surfaces, which could be different wettability. Black silicon is not super hydrophobic. The chemical composition could be different. The nano protrusions or nano features of the surface could be also different. Bactericidal effectiveness could be also variable depending on the pattern. However, what you see here, that sometimes synthetic analog of natural bactericidal surface may achieve a greater performance than the natural template of bactericidal surfaces. So it's all about optimization of the surface nanoscale parameters. The mechanism in the context of dragonfly is not really defined as yet. We believe this is our hypothesis. It's still stretching of the membrane when it's ruptured in between the nanopillars, because even the black silicon nanopillars appear to be sharper. They are not reaching the dimensions below 10 nanometers. So basically, it's very, from theoretical point of view, it's very difficult to pierce the cell still. The membrane will be hard to pierce. What is another remarkable thing about this type of the surfaces? In a way, they are self-cleaning, but they are not self-cleaning as it was in the context of the water when it's bouncing off the surface. But it is self-cleaning when you see that the cell debris actually removing from the surface over the time. The removal of the cell debris from the surfaces is not as quick as it's rupturing, and it's highly dependent on the nano pattern, right? But in a range, it could be 20 to 30 minutes, which is required to remove the dead cell from the surface, where the debris is probably floating around in the environment. Now you may ask, how about eukaryotic cells? What happens with eukaryotic cells? If bacterial cells are effectively ruptured, what about big, large cells? As you can see here, that was our work some time ago, where we used fibroblast-like OS7 cells. What you see here, it's a movie of attachment and settling of COS7 cell on the black silicon surface. And here you see a freeze fracture image where you see the cell and the nanopattern, right? So the cell is safe on the nanoscale pattern. Well, if you think from the logical point of view, the nanopillars are too small. And I'm always saying that perhaps it's like a mosquito bite for us. That's what the nanopattern for eukaryotic cells. It's a matter of different scale of the pattern which may affect. So for eukaryotic cell, bactericidal surfaces are safe. And in a way, it has a very significant, and it's very important in the context of implantable biomaterials. Well, what you see here, it's another experiment. When we try here to infect the surfaces with infectious doses of pathogenic bacteria, and after that seed these cells, these surfaces with eukaryotic cells. And as you see here, within 6 hours, bacterial cells are effectively ruptured, and eukaryotic cells, you can see on the next slide, are growing very nicely. So what you see here is the COS7 cells on black silicon pre-infected with Pseudomonas aeruginosa, and the same surfaces pre-infected with Staph aureus. And you see on day 7, eukaryotic cells are growing nicely and reaching confluence state. And on the surfaces which are flat, or the ones which are used for implants, the infected surfaces keep going with the bacterial cells, pathogenic bacteria happily growing on these surfaces, and they in fact inhibit and contaminate eukaryotic cells, and eukaryotic cells are not survived on these surfaces. Moving on, the effect of mechanobactericidal surfaces, as we are trying to now develop it further, to show you that it's quite versatile. So there are different ways which we can use, or we can use a different type of nanopattern to kill bacterial cell by a different way. This is another example when we can cut bacterial cells through using the nanostructured surface. And this surface could be a graphene-like surfaces. What you see here, that you can have a graphene surface with the graphene flakes, which is very easy to obtain. And we looked at different types of the flakes fabricated from graphene, and see how bacterial cell will respond on this. On the top you see graphite surfaces, right? And these two types of graphene surfaces, they in fact highly bactericidal. But until now, the nature of bactericidal activity of graphene flakes is not really resolved. Well, there are two hypotheses. One hypothesis says that extraction of the bactericidal effect of graphene flake, extraction of the lipid from the membrane, that's what they say, what happens when the cell interacts with the graphene flake. What we think it is, and that is our work where we're trying to prove it, that actually it's a pore formed due to reorientation of lipid tails to graphene surface while they're interacting with the graphene flakes. So what you see here, it's a little bit enlarged images, confocal images, showing the red cells which are killed looking like swollen. And that's apparently because the cell, when it's cut through, the environment, external liquid comes inside and it appeared as a swollen. Now, the theoretical analysis were done with collaboration, the same theoretical physicist group in University of Ravira and Virgili. That is Vladimir Baulin, you see here. And they apply single-chain Mail field simulation to show that the pores are actually formed at the interaction. So we believe at this type of bactericidal activity, the bactericidal efficiency of graphene-like substrata or graphene-like materials of this kind depends on the lateral size, shape, and interactive angle of exposed sharp edges, which are likely to puncture the bacterial cell membrane. Another way we can kill bacterial cells using nanostructured surfaces is this. I'll show you how bacterial cells are teared apart on the nanostructured surfaces. What you see here is exceptionally high aspect ratio, up to 3,000, of vertically aligned carbon nanotubes, right? So this work was done in collaboration with group of Michael de Volder in Cambridge University. So what they've done, they grew carbon nanotubes from a catalyst layer, and which was deposited on a silicon wafer using physical vapor deposition. And we used two types of the pattern with the 1 micron and 30 micron vertically aligned carbon nanotubes. What you see here is that this type of the surfaces is also quite bactericidal. The highest bactericidal rates of 99.3% for Pseudomonas aeruginosa and 84.9% for Staph aureus were recorded on 1 micron tall vertically aligned carbon nanotubes. Now the question is again how this happened, why? And this is quite interesting. The same group, in collaboration with the same group, what we found, and they applied so-called engineer's beam theory or linear theory of elasticity. The same theory was applied for engineering of Eiffel Tower and this large wheel. It's exactly the same theory used for construction, and we apply it for to explain the effect of exceptionally high aspect ratio. So what you see here and what is explained, it's just a stored and released energy. So basically the bending, very tall nanofeatures, the bending, they store energy, and when they bend back, they release this energy, and that's how they tear the cell apart. And it's quite logical to imagine that, of course, the 1 micron nanotubes actually more rigid and they store higher energy, and that's why they are more effective. And I think I just would like to finish up with some aspects of the nanofabrications, what is currently in the field, and give you just one example of our recent work on hydrothermal treatment of titanium. So basically there are different types of nanofabrication techniques which could be applied to produce nanopattern, and the nanopattern could be quite different, right? It still will be active, but the range of the activity could be quite different. So that is a challenge. And these are techniques you can use: laser irradiation, lithography, plasma etching, electrospinning, and even simple, that is a nano-implant lithography, template methods. It's all about the resolution, but with advances in nanofabrication where we can reach nanoscale resolution, then the results will be always promising. Now, the hydrothermal treatment is one of the examples of nanofabrication using a very simple technique, but again, it's a matter of optimization of parameters and time of the treatment, where you can fabricate the surface of different patterns. So what you see here is the formation of asymmetrical nanosheets with sharp nano edges. And in a way, I would say that the mechanism of bactericidal efficiency here could be similar to the sharp edge of graphene nanoflakes. It's in the same type of bactericidal effect as we believe it is. If you look at the activity, bactericidal activity, you see that the Pseudomonas aeruginosa could be like up to 100% cells killed in a quite large range of the treatment conditions. However, Staph aureus is much more difficult to kill, as always, but it could be high rate of killing achieved after 6 hours treatment. of the surfaces. And I would like to finish up again with this particular antibiotic-resistant problem and show you if nanostructure surfaces, and we call them mechanobactericidal surfaces, can kill antibiotic-resistant strains. So what you see here, I just wanted to emphasize that we used, and that is a report from Centers for Disease Control, again from United States, 2013, and this year in autumn they will produce a new edition of the same report. They identified top 18 antibiotic-resistant threats in the United States, and methicillin-resistant Staph aureus is one of those threats regarded as serious threat, right? What I also found quite upsetting data that in a way that why it's epidemics, right? That in our community, it's up to 5% of healthcare workers in the United States are carrying this methicillin-resistant Staph aureus, and up to 2% of the population actually carrying the same thing. Now, I probably wouldn't stop here. Just to tell you that the antibiotic resistance mechanisms are quite different for different types of antibiotics. In case of methicillin-resistant bacteria, what happens with this strain? First of all, the drug cuts the bonding, the cross-linking, that is again peptidoglycan. So once the drug cuts this cross-link between the peptidoglycan layer, network is damaged and the cell is dead. What is doing the methicillin-resistant strain? It's actually rebuilt the bonding again, and that's how it survived. That's how the resistance is developed. And what we did in this work, we used methicillin-resistant strain and methicillin-susceptible strain. This is the original standard strain. And we put this strain on the hydrothermally treated titanium surfaces. And what you see here, they are dead. They are dead. Doesn't matter. In the presence of antibiotics, I should say that the build-up of this reconstruction of the bonding in the peptidoglycan layer triggers by the presence of antibiotics. That's why we run experiment in standard physiological condition and in the presence of antibiotics. As you can see, both types of the strains are successfully killed by the surface. So yes, the conclusion, we arrived to conclusion that highly bactericidal nanotopologies of insect wings represent the first reported example of purely physical antibacterial activity and opened a new era of biomedical antimicrobial nanotechnology. These mechanobactericidal surfaces will be potentially useful when applied to implants, reducing risk of post-operative bacterial infection and minimizing the demand of antibiotics, thereby helping to combat antibiotic resistance. And mechanobactericidal surfaces, of course, can be exploited in various industrial and bionic applications. Future directions. So the target now to fabricate the surface with a dual functionality. So they will be repel bacterial cells, and the ones which will not be able to repel and settled on the surface will be killed by the surface. This is our next goal, and hopefully we can achieve it with the help of good students. And I think that was all from me. I would like to acknowledge many people. Without excellent contribution of these people, this work won't be finished, confirmed, performed. And of course, Professor Russell Crawford, Khan here, not here, lots of people from Swinburne University of Technology and colleagues all over the world. And that is the last slide with the students. And was hoping that the movie will run showing the work of the students in the lab, but looks like it's not. But thank you very much for your kind attention.
Thank you very much for a very interesting and entertaining talk. Any questions? We can have a few minutes for some questions. Yes.
So did I go too long?
Just about right time.
So, beautiful talk, eye-opening. So, one potential problem, one potential opportunity. In terms of lysine or targeting other type of lipid-enveloped organisms. So pox viruses, the largest viruses, around 500 nanometers, and they're lipid enveloped. So I was just wondering, as you were talking, if there might be some utility also in that area. And the problem, the cells, the eukaryotic cells that at least you've tested in this presentation, they have very nice mechanisms for recycling their membrane, for keeping membrane stability. But red cells, we have a red cell expert sitting there at the front, do not have those same capacities. So how would you see first the opportunity for the pox viruses and perhaps the in vivo utility in terms of perhaps negatively affecting red cells?
That's a very good question. I'm impressed you noticed the possible effect on the red blood cells. You are absolutely right. Red blood cells are very specific and they will not be safe on the pattern. And we actually did this work and we showed that they are not safe. But again, what I want to emphasize all over again, it's a matter of the pattern. So you can optimize the structure, the nano-features of the pattern, and use the pattern which will be bactericidal to bacterial cell, say even the super high aspect ratio features, which can kill bacterial cell by a different way. But by this way, the eukaryotic cells will remain safe again, including red blood cell. We simply didn't test it. But that will be a beautiful work, actually, to compare different types of the bactericidal pattern and see how they affect red blood cells. But I think that's not all pattern will be similarly affecting red blood cells. As for the viruses, it's a matter of, again, nanopattern and the fabrication. I remember when we just started working on this, the resolution of the nanopattern 7 years ago was so poor that we couldn't even dream about what we can do now. So for the viruses, you need the pattern with more fine features, nano features, to see, but I can't guarantee. So it needs to be some work done.
Okay, thank you very much.
Thanks for such a fantastic presentation and beautiful work. I'm also wanting to follow on from Magdalena's question about how you think this technology might work inside the gastrointestinal tract to engineer the microbial population, so really targeting and dealing with that mucus, biofilm, and that sort of environment.
I don't have any answer on this question because no work was done. I don't know. There is no perfect solution, but because that's the nature of science. As long as we go on our way, we find something else. So far, I don't know the answer, but I'm sure that we can handle that in future and find a solution for
and we
it.
can discuss together.
Maybe just one more question. Can you put these surfaces onto catheters, on plastic surfaces?
Yes, it could be. Like I said, maybe 5-7 years ago, it was basically impossible to put this particular nanoscale resolution in the range of 10 to 100 nanometers on the plastic. Now it is possible. I can tell you that there's a company in Japan. They are very close to fabricate that type of the nanopattern in a large commercially upscaling area. So yes, it is coming.
Thank you. Do we have any more questions? Yes, okay, you can have a second.
So I went to a beautiful talk recently by Professorial where she was describing on the smaller scale how polymers, by their nature and flexibility, could avoid the binding of proteins, and how their movement and their topography was very, very important. Now, as you were talking, I was wondering, because you were saying there's the issue of the attachment, then there's the issue of the clearance. Mm-hmm. And I was wondering, when you were presenting your talk, all the surfaces you were showing us were rigid, and whether there were any plans of also exploring motility.
Yes. And it is also work about to be completed. So, like I said before, it was very difficult to run a systematic study with changing only one parameter on the surface, like a length of the nanofeatures. It could only was possible to achieve using nanolithography, which is very expensive and very kind of difficult to achieve. We still managed to have this particular type of the samples and about to finalize this work, where indeed the bending could be a part of the effect. Yeah.
All right. I think we might just have to stop here. We got the refreshment next door, so you can continue to talk to Elena and mingle each other. So thank you very much for coming, and hopefully we'll see you next time. In September, we'll have another one. So thank you very much.
Thank you. Thank you. Thank you.